All right, so we want to be able to do large cohort analyses. We want to be able to scale up to increasing sizes of samples in order to have better power for analyses. And we want to be able to add samples incrementally over time without redoing a lot of the compute that we've already invested time and money in. So there are two tools that are involved in this part of the workflow. HaplotypeCaller is used to identify potential variants in each of the samples, and it outputs per sample genotype likelihoods in an intermediate file format. And then as the second part, we're going to combine together all these intermediate files to do what we call joint genotyping on the entire cohort using the genotype GVCF tool. So starting with HaplotypeCaller, you can kind of break the algorithm up into 4 different steps. The first step is to identify active regions. Most of the genome looks pretty much like the reference, so we're only going to focus on areas where we think there might be variation. The next step is to assemble plausible haplotypes. Then we're going to evaluate each of our reads against those haplotypes to determine the per read likelihoods. And then finally, we're going to genotype each of our sample against the discovered alleles. So the first step in identifying active regions, again, we don't want to invest a lot of compute in areas that just match the reference. And given that you see a SNP about every 1,000 bases in the human genome, there's going to be a lot of areas that are just reference that we can skip over. So what we do is we take a sliding window along the reference and we look at the reads corresponding to that window. We count up the mismatches, indels, soft clips, and just generally indications of variability within that window. And we use that as a measure of entropy. And so effectively, what's going to happen is we do like a rough pile-up genotyping at each of these sites. And based on the variant or the genotyping call at each of those sites, you get either a value that's very near 1 or 0. And so each of those positions there that feature alternate reads will produce that little spike in the measure entropy function that we're going to use. And then we smooth that over by convolving it with a Gaussian, if you're familiar with signal processing, so that we can have a wider window to take into account all the evidence that might inform that region. And so down below, we're showing you the smoothed version of that signal. And then we establish a cutoff. And if the value of the function is below that cutoff, we say it's inactive. It's not worth computing on. There's no interesting variation there. And then we use the other cutoffs to determine the bounds of our active region that we're going to continue with into assembly. So we do local assembly with respect to the reference. This is not de novo assembly. We are not going to discover large events, large rearrangements. Again, we're only looking at a small window, and we're constrained to mapping these things with respect to the reference. So the first thing that we're going to do is add the reference sequence to the graph, and then we split up each of our reads into short k-mers. You know, where like a 2-base pair sequence would be like a dimer or a trimer for 3 and so on and so forth. We call it a k-mer if it has k bases in it, where that value can vary, which helps us take into account regions of high repetitiveness. And so we add those to the graph based on places where they overlap with the reference and the existing paths. And then once we have added all those paths to the graph, we collect the haplotypes, and we align those to the reference using Smith-Waterman to find out what the events are. So we'll get into a little more detail here. This is an example of an assembly graph produced with the HaplotypeCaller. And basically what I did here is I only put in relatively short sequences to represent reads and one of each. So in your real data, you would see a lot more reads, a lot more nodes, and these numbers here are meant to indicate the number of reads that have been seen on each of those paths. And so those are a little low in this example, again, because it's a toy example. But for example, what we have is we took the reference first and we chopped it up into k-mers of base pair 5 and 5-mers. And so the first subsequence of our reference is this GTCTC, and that's where we're going to start. And then the next 5 bases of the reference are going to add on this A. And so the way we're representing it here in the graph is each of those single letter nodes are the new base that we're adding, and then the previous bases from the other nodes until you get to that 5-length sequence. And so the blue pair here represents the reference, which is how we're going to start. And then for each of our quote-unquote reads in this toy example, we chop those up into 5 base pair segments and add them to the graph. And here you'll see that this k-mer didn't overlap with any of the parts of the reference because it has that alternate A. And so it's separate here. Even as we add another A onto the end and we shift that window by 1, you're still going to see an A, C, T, C, A sequence, which doesn't match with the reference. So it's still a separate path in the graph. And then we add a C onto the end. So at that point, your 5 base k-mer is going to be CTCAC, and now both that read and the reference match, so we can merge those paths back together in the graph. And by similar logic, when you see sequences that have a different base, they're going to split off from the reference path. And so this graph represents all of the sequences that went into it, but show you how the reads had large pieces of the sequence in common. And it becomes a little bit easier to pick out the points of variation, like here there's a T to A, or an A to T SNP. Here there's a C to T SNP, C to G, T to C, and so on and so forth. So we can traverse the graph now from those initial nodes, following each and every path to get the candidate haplotypes, which are enumerated there on the right side of the graph. And again, the numbers on the graph are meant to show how many reads support each of those paths. Typically, in a real example, we would have a lot more reads, and we would trim out any paths that had fewer than 2 reads supporting them because they're likely to be an artifact, and it's going to increase our computation if we have too many haplotypes. But again, we just had a small sample of reads here. So the reason that we do graph-based assembly rather than just relying on the alignments that BWA-MEM gives us is because it's able to recover some indels that get represented by BWA in a way that's not really a consensus of all the reads. And we're able to remove a lot of artifacts. So for this particular region from a real NA12878 BAM, we see that in the BWA-aligned version, there are these sequences that don't really fit where they are. They have a lot of soft clips, and there are a couple different representations here of indels. Some of them have a 12 base pair deletion. This one has a 3 base pair deletion, and then a couple matches, a 7 base pair deletion. And then I think there are some 8 base pair deletions here and 110. So it's a lot of evidence. There could be multiple events there, but it's really hard to tell. And that's because each read was aligned individually without using information from the other reads. And so depending on what the start and end position of your reads are, the best alignment can look different based on the context of reads. But when you look at all of them by assembling them through the haplotype graph, then you can have more of a consensus call. And so once we run this data through the HaplotypeCaller, we're showing you the BAM output, which is an additional option you can add to your command line that'll produce a small BAM with the realigned reads. So here we're showing all the haplotypes that were recovered from the assembly graph, kind of like what we just looked at in the previous slide, and then the reads aligned against those potential haplotypes. And so you'll see that there's potential for a couple different events. We're still seeing a 12 and an 8 and a 10 like we did in the above read. But once we reassemble all of our reads and align them to those haplotypes, now we're seeing a much stronger signal that there seems to be 2 different deletions here, one of which is 12 bases and one of which is 10 bases. And so from that really, really messy BWA-aligned BAM at the top, where we weren't entirely sure what was going on, we're able to see that it seems to be, at this position, there are 2 different deletion calls. But they're in pretty good allele balance because we see about 50% for one, 50% for the other. And we see actually 2 different lengths for those deletions. And again, because we're using the consensus, we're going to represent all of our reads in the same way. So for whatever reason, in these original BWA alignments, this particular sequence for which the reference here, this path on the top with the blue, would have been a TTA. Instead, we see a TACA. So that you could either represent that as an insertion of an A and then the T changes to a C, or the T changes to an A and then there's an insertion of the C at the end. And depending on the span of the read, like the start and end positions in the context, BWA represents these differently. And so again, the benefit we're going to get going through the assembly graph is once we have the haplotypes that we recover and we align all the reads to the same set of haplotypes, it's going to form a consensus across those reads and give us a cleaner call. So another one of the benefits that we get out of HaplotypeCaller is having physical phasing of the variants within these active regions. So it's within the span of an active region, which is only a couple hundred bases, but it's still very useful from biological context. So on the left IGV screenshot, we're showing you a pair of SNPs. And in these SNPs, the variants are on the same haplotype, whereas in the other screenshot, the variants are on two separate haplotypes. And so that's kind of it's the difference between having a gene that has one copy that's normal, one copy that matches the reference, and the other one has two potentially missense mutations, versus having each copy of the gene having a missense mutation and not having any copies that are functioning as they would, as you expect in the wild type, which is a very different biological situation. And we annotate these in the VCF with our PGT for phased genotype and PID. So the PGT tag here is going to give the position on its contig and the reference allele and the altered allele. And then the genotype then, here we're showing phase version that these two are in cis phase because the zeros are on the same side of the pipe as the ones. Whereas if you saw one in trans phase, one of the entries would be 0 pipe 1 and the other one would be 1 pipe 0 to show that they're not on the same haplotype. So functional implications on the short scale, having the phasing information could turn two separate haplotypes with missense mutations potentially into one normal haplotype and one haplotype with a loss of function, turns to a stop codon. But you could also have functional implications that are much longer range. For example, if those missense mutations change the charge on an amino acid, you might have had normal amino acid that has an affinity for an amino acid on the other side of the protein. But then if you have within cis, maybe they both change charge and they still have physical affinity for each other. But if one of them changes charge and one of them doesn't, in either way, that could potentially cause conformational consequences in your protein that it codes for. So focusing still on this small active region of a couple hundred bases, we have done the local assembly. We've identified the candidate haplotypes that we think describe the variation in this region, and we're going to take each of our reads and find the likelihood that it was derived from each of those haplotypes. And so we use an algorithm called the pair hidden Markov model, or PairHMM, to do this. And it's a little more advanced than a Smith-Waterman alignment that you might be more familiar with because it incorporates the base qualities also, which is important because we want to know, given that we have confidence estimates on each of the base calls in our reads, that should be able to give us a better informed likelihood of how well that read supports each haplotype. So this is getting a little more technical about the PairHMM to show you the states that are involved. If you run GATK3 in the classic way, such that you are outputting base insertion and base deletion qualities, they help inform the transition rates in this diagram. So I'll tell you a little bit about the states in the diagram. What we want to do is align two sequences to each other and allow for one or the other sequence to have extra bases with respect. So in the case of the reference having extra bases, that would be a deletion in the read. In the case of the read having extra bases, that's a typical insertion. And so as we iterate through positions looking at each one of these sequences, it's going to be in one of three states. Either they're going to match each other, which here we're not requiring that the bases have to be identical. Like we could still have a SNP in the match state. We're just saying that neither one of them has an insertion with respect to the other. Or you could be in what we're labeling here as the IX state, where there's an insertion in the reference with respect to the read, just a deletion in the read, or in the IY state, an insertion in the read with respect to the reference. And so then these gap open penalties so the gap open penalty is going to come from those base insertion and deletion qualities if you're using those. If not, we have a constant factor that we've validated empirically. And the base qualities are going to inform this probability of staying in the match state. And so if you really want to dig into the details here, I highly recommend Richard Durbin's book called Biological Sequencing Analysis. I think there's a citation at the end of these slides. But it talks a lot about the alignment of two sequences using Smith-Waterman, more advanced affine gap versions of Smith-Waterman, and the PairHMM. So after evaluating this incredibly computationally complex model for many, many reads against a couple of haplotypes, we end up with this matrix of the likelihood for each read with respect to each haplotype. And so we're going to take that matrix into the next step in order to do our genotyping. So we've filled in that matrix now with some toy values. Again, the numbers that you see here are nice, large likelihoods that are easy to visualize on the slide, but in reality, you're going to see much lower numbers if you ever decide to debug those values. So what we want to do is go from haplotypes to events, right? Because each haplotype can vary from the reference in more than one way, potentially. But in the VCF, we need to enumerate events that happen at a site-by-site level. So for example, the haplotypes that we're looking at here, we're showing the bases that match the reference in red, and we're showing the bases that are different with a red box around them and a white letter. So there's both an A to C SNP and a to G SNP if you compare haplotype 1 and haplotype 3. So there are two events here and two different combinations of things that can happen in each of those events, which gives us 4 haplotypes. So if we want to look at just that first event, there are 2 different haplotypes that contain that event. So we need to decide how to determine the likelihoods of each of the reads with respect to the A allele in that first variant position or the C allele, which is variant. So we take the maximum likelihood across the haplotypes that contain that event. So for example, for read 1 across the first row, if we look at the C allele, the C is featured in haplotype 2 and haplotype 4. So we're going to look at the best values from haplotype 2 and haplotype 4, and it's 0.1 from 2. So we copy that value over. It's encoded in green, so you remember which haplotype it came from. And then we do the same thing for the second allele at that position. So allele A is seen in haplotype 1 and haplotype 3. If we compare those values, we see that the greater value comes from haplotype 3, so we copy that value. And then we do that for the same in the read, and that's the way that we can break down these haplotypes, which may contain many variants, into a site-by-site likelihood matrix. And so step 4 is going to be outputting the genotype for each sample at each of these sites that we've now broken our likelihoods down into. So we want to determine the most likely combination of alleles for each site. We're going to go through diploid genotyping examples because that's what we see in humans, but it's extensible relatively easily to organisms with higher ploidy. We do support higher ploidy calling with HaplotypeCaller. We've had people do it all the way up to ploidy 100, which is one way of dealing with mitochondrial data. So it's a little intractable in terms of the output, but it is computationally possible. So again, we're going to do this genotyping with respect to the allele likelihoods matrix that we just broke down. And we're going to apply Bayes' theorem with a diploid assumption for humans. So, so this matrix here is going to represent the likelihood of, and excuse me if I confuse my likelihoods and probabilities because I was raised frequentist, but I'm learning. But each of the values in the matrix represents the likelihood of the haplotype given the read. So the reads are the things that we get to observe, and they are telling us about the haplotype, which is what we actually want to know about our sample. It's not interesting what reads you have in the sample. You want to know what does the sample's genome actually look like. So in order to evaluate each of the genotypes, we look at each combination of haplotypes And again, we're looking at an allele by allele basis. So for this example, the A is actually the reference. So for a homozygous reference genotype, we would look at the likelihood of observing two A's. So we can say H1 is the allele A, H2 is also the allele A, and then we can plug into this equation the likelihood of seeing allele A given read J as we iterate through all the reads. So we're going to fill in these values with the A entries from our matrix for each read. In the homozygous reference case, those two are both the same, so the denominator collapses. And then we just take the product over all the reads to get that likelihood. We do the same thing for the heterozygous genotype, where we have one A and one C. In that case, we're going to fill in different values for these for these entries in the equation based on our matrix. And we're going to do this also for the homozygous variant genotype. So again, we're not taking into account phasing in the genotyping. So an A/C genotype, same as a C/A genotype. And then in theory, we take a posterior probability of the genotype, because what you're going to get out of that equation are a bunch of likelihoods, and likelihoods are not guaranteed to add up to 1 in the same way that probabilities are. So we need to get posterior probabilities out of them, and we apply Bayes' rule. And at this point in the game, we're only going to apply a flat genotype prior. So we're going to say that each genotype is equally likely. We'll look in the genotype refinement workflow at a better way to inform those priors later on. So as we go through these steps with our real numbers, we compute each of these genotype likelihoods, as I explained, for the different combinations of alleles. And sure enough, those three likelihoods, even though they encompass the full event space, don't add up to 1. Again, they're likelihoods. They don't follow the same rules. But once we run it through the Bayes rule calculations, that's going to help us normalize our likelihoods so that we get posterior probabilities that add up in a way that we would expect. So across these probabilities, our highest is for the genotype CC. So that's what we're going to assign in the VCF, and we're going to output those probabilities as Fred-scaled likelihoods in the PL field. So we talked a little bit about Fred scale already, but we'll show you how the conversion happens between the 0 to 1 scaled probabilities and the Fred scaled values that end up in the VCF. So our highest probability was for CC, right, which is our homozygous variant genotype in this case. So we take each of those probabilities because we always output the PLs in the same order with home ref first and then het and then home var, and in a similar way for multi-allelic sites. So convert those probabilities to Fred scale, again with that -10 log 10 conversion we looked at. Normalize such that the most likely genotype ends up with a value of 0. So just subtract off 2.46 in that case. We round to integers, and then those become the PLs that we report in the VCF. And then in order to get the genotype quality, that's a description of our confidence in the called genotype versus the second most likely genotype. So the called is going to get a 0. So we look for the largest non-zero value to be our confidence. And so, for example, this A to T SNP in the format field, we'll be looking at genotype quality, and Fred-scaled likelihoods. And here we have 1, 1, and 3, which is our genotype quality, and then that whole array of values as we computed above. So we started this process with an aligned, quality recalibrated BAM with duplicates marked that's ready for variant calling. We input that to HaplotypeCaller. We identified what we call active regions, where there's enough information about places that vary from the reference that it's worth doing a little more computation. We take the reads from those active region areas and align them to the reference to recover potential haplotypes. We evaluate the likelihoods of each of the reads coming from our potential haplotypes using the PairHMM to determine the per read likelihoods. And then we use those likelihoods to get a genotype call at each site that's variant in the sample. And at the end of this process, we output a VCF. So we skimmed over a little bit of the detail for the joint calling exercise because we went through a single sample, but ideally you want to be able to analyze multiple samples to improve your power of variant discovery and analysis beyond that. So what's going to happen is we're going to output these intermediate GVCF files, where the G stands for genomic, because it's giving you information about every genomic position. So we're going to run HaplotypeCaller in GVCF mode, or emit reference confidence, to output a GVCF. And then this is going to give you these genotype likelihoods for each one of your samples. And then we're going to run genotype GVCFs over all of those samples using a multi-sample variant calculation or variant confidence estimation model to do the final joint genotyping. So how are these GVCFs different from a traditional single sample VCF? In order to capture information about variants that weren't seen in this sample, we're going to add this non-reference allele here, which has got these little less than greater than brackets around it because it's a symbolic allele. It doesn't stand for a particular sequence. It just stands for anything that's not represented in this line of the VCF. And we do this because you're going to see variants in some other sample eventually if you keep adding together all the samples in the world. And you need a likelihood to assign to that because, for example, this sample that we're looking at here in this first blue line had all reference. But if you sequence it together with another sample that you want to joint genotype, perhaps that sample has a T, now you need to go back and fill in the values for your original sample, like what is the likelihood of having seen a T in that sample, which should be low, right? Because we didn't see anything that made us believe that it was non-reference, but we do still want to assign a value to it. So we have this non-ref placeholder allele, which gets a likelihood based on the base qualities, which are going to give us an indication that our reference calls were wrong. And then we have a heuristic model for the possibility of an insertion based on the context of the reference. So basically trying to put into one numerical factor the possibility that there was an event there, we just didn't happen to see it in this sequencing for this sample. So that's describing this start position at about 1,204,000. And note that there's also an end tag because in order to save space, we want to be able to represent a whole bunch of reference lines in the VCF in one block. Because otherwise you're going to end up with a 3.2 billion line VCF for your genome, and that's just not easy to work with. So instead we compress all the blocks that are called as homozygous reference in that sample, and we give the end of the block with the end tag there. And then if you do see a variant in the sample, it's going to look a little more similar to what you would expect from a single sample VCF, where we have the reference allele here, the discovered alternate allele T, but it's still going to have this non-ref because we want to take into account the fact that in some other sample you might still see something that's not a C or a T. So once we combine that GVCF intermediate file with some of our other samples from the cohort, we use this genotype GVCF tool, and that's going to output a multi-sample VCF. So see here, this is just for NA12878. But if we combine her together with some of the other samples from Cells and Genomes Project, now we have 3 samples, as shown by these sample names in that line of the VCF, and we'll have 3 genotypes. And so already we see that there was this variant discovered at position 1,117,000, where we didn't see anything in NA12878, but what we'll do is we'll take her values from the non-reference case to apply likelihoods to those alleles. And for example, here we have the C to T that we did see in NA12878, and she has a heterozygous genotype as we would expect from the above VCF. And that was also seen in the other samples that we sequenced. So as I mentioned, GVCFs are genotype or genomic VCFs. because they give you information about every position in the genome. The example that we just looked at has these non-variant blocks or blocks of reference information where they're going to have a start and an end tag to tell you how many bases they span. But if space is not an issue for you because you're doing maybe like RAD-seq or something like that where you have a very small number of bases that you're focused on, you might want to gain a little bit more precision in your genotype likelihoods by doing base pair resolution, in which case you can have a similar format, but you do output an entry then for every single position, even if the sample's referenced. So you'll get separate records for each of these homozygous reference calls in your sample. Likelihoods will be very similar, but a little bit more precise. So here are the commands for how we run this tool. Again, you're going to run HaplotypeCaller on each one of these BAMs separately. And this is why it's nice to have a workflow, because rather than running each of these commands or putting them all together in a shell script, you can then set up your workflow and send it a list of sample names, which is wonderful. And so it'll run HaplotypeCaller for each of those samples. Each of those BAMs go in, and then a GVCF comes out. You'll note that we name it with a .g.vcf so that you know that it has the non-reference allele. It's not ready for analysis yet. And we added this emit reference confidence, GVCF argument, to say that we do want the non-reference allele. We want to combine this with other samples later. And then once we run HaplotypeCaller on all the samples that we want to use, we can run genotype GVCFs to combine their intermediate files together to get one final call set. So there's a note here that if there are more than 200 samples, typically we do them in batches with combined GVCFs, which is our GATK3 tool. In GATK4, we're moving on to using a product that Intel developed for us, which is called GenomicsDB. And it's a more efficient representation of a set of combined GVCFs. So that's really helped us speed up our production pipeline. We were struggling calling 20,000 genomes together in one call set because there's just so much data to put into one place. And so GenomicsDB is a much more efficient way to represent those intermediate files when you combine them together before you output your final genotype VCF.